So I'm Alice, and I'm happy to be here today to tell you about some of the clinical applications that I've used the nanoimmunoassay. I'm currently an instructor in oncology waiting for my assistant professor appointment to come through, hopefully next month. And in medical oncology, we have many needs for being able to look at high resolution of proteins. I mean, if you think about a cancer patient, there are periods of opportunity that can, to make assessments that can really help us make clinical decisions. First, something happens to initiate the tumor in the patients, and often this is way too early for us to know that anything's happened, and you have to wait till it grows big enough to be able to make a diagnosis and do staging. So example, for lymphoma, you get a chunk of tissue from surgery or a fine needle aspirate, you do a tissue examination with histology, immunohistochemistry, and flow cytometry for lymphoma, and do a gross evaluation of the tumor burden, which is a physical exam because you can feel lymph nodes, but also CT scans and PET scans to find all the places in the body where the tumor is, figure out what stage they are, and then put all this information together to choose your best treatment. Once you start a treatment, something happens to the cells, but this is where we don't really have the luxury in patients of knowing what's happening to the cells, whether the drug is actually getting to the target and to what level it's lowering the target if we're trying to inhibit a pathway. All we do is we treat for 3 months, and then we evaluate for a clinical response, which basically is re-examining the tumor burden with imaging. And after 3 months, either it's worked because things have shrunk, or it hasn't. And it's really sad that if it hasn't worked, the patient asks, well, why not? You know, did it not get there, or what's wrong with my tumor? And we just don't know because we have no way of really figuring out what's happening during this period, which is really critical, not only for drugs that we give to patients now, but also as we're thinking about developing new ones. How can you develop a new drug and not know what's happening here in each patient? So a lot of biomarker work right now is focusing on this step because this is when we can get the most tissue. For example, breast cancer, they've gone to surgery and you've got the whole tumor out. And a lot of beautiful work is done at the DNA level, looking at deep sequencing and things like that. And RNA, we can do PCR, which is very sensitive, microarrays, which is high throughput, as well as now RNA-seq. But much less is being done with proteins. And I know this is the focus of this session, which is wonderful. And if we think about it, the gold standard is a Western blot. But we know, everybody in the room that's done a Western blot knows how variable that can be just based on the many manual steps, as well as the fact that it's only semi-quantitative, as I'm sure Joanna talked about. And it takes about 1 million cells, which is prohibitive if for some of our clinical samples, all we have are a fine needle aspirate, which can be 10,000 cells, or if we're trying to look at something like circulating tumor cells, which is like 5 to 10. So our gold standard really needs to be improved upon. Flow cytometry is awesome, and you're going to hear, I think, the next lecture talking about single cell analysis. But it requires a single cell suspension, which we don't necessarily have that luxury for a solid tumor. If you digest the tumor, it could really change the signaling by the time you get it onto the flow cytometer. And then mass spectrometry and things like that, which again right now are mostly requiring large amounts of input. And of course, coupling the two, which you'll hear about with the CyTOF. And if you think about it, though, it's really the proteins that are the army that carry out the cellular functions and tissue functions. So changes in RNA, even if you've done a microarray and found 10 exciting hits, you always validate it at the protein level. And especially for cancer, when a lot of our new therapies are inhibiting kinases and enzymes, you really want to look at what's happening downstream at the phosphoprotein activation level. And so that's not something you're going to see by sequencing. So this is where we really want to start building in additional time points leading up to just a gross examination of tumor burden with imaging. And this is kind of what I'm going to be focusing the rest of the talk about. So when I say we want to determine that red box, the biologic response to treatment, I'm talking about profiling changes in cellular pathways of tumor cells or host cells that occur after administering a therapy. So for example, I wrote a clinical study, and we built in clinical sampling where you do a fine needle aspirate of the tumor prior to starting treatment. You start your daily treatment, and then day 8, which is based on preclinical studies of when you expect to see decreased signaling and increases in apoptosis, you take another sample of the tumor and assay for biologic response. And this is what I've been using the nanoimmunoassay for, to look at the proteomic level. And then you correlate that to the clinical response, which is standardly done at 3 months, and try and get an earlier determination of what's happening at the molecular level. And so the barriers to this we've talked about are the amount of specimen. I mean, if you've got a patient who's got, say, metastatic disease and they've got a tumor there, and you want to go back in and get another sample, it's not really feasible, and you wouldn't put the patient through another surgery to get enough tumor tissue just to do a Western blot, especially if you wanted to look at 100 targets. That's a lot of tissue you need by Western. We really need a very highly quantitative and sensitive method so you can get away with a really small sample. And also, when we're talking about looking at phosphorylations, it's really hard to measure multiple phosphorylations with other technologies. Even Western blot or flow cytometry depend on having a phospho-specific antibody, and you don't always have those, and then it's really hard to control for background staining, especially at the single cell level. But with nanoimmunoassay, we can really start addressing some of these barriers. And so I've used the high-throughput automated NIA for charge-based separation to profile cancer signaling pathways in patient cells before and early during treatment. So this is some preclinical data that leads to the clinical study, the first one I'm going to talk about. And that is that we found that statins induce lymphoma regression and apoptosis in a preclinical transgenic model of lymphoma. This is work done that was a collaboration of the Felser lab, the Nolan lab, and the Steinman lab. And what they were studying is that statins, which are HMG-CoA reductase inhibitors, which are cholesterol-lowering agents that many people in the room may know about, well, how they also work is they can inhibit ERK, AKT, and STAT3 phosphorylation in T cells and lymphoma cells and immune cells. And so what we found in the mice is if you treat these mice with lymphoma, as early as day 8, you can see that the tumors shrink. If you look histologically, there's evidence of apoptosis, which we confirmed with tunnel staining, which are the bright green cells. And these are the things, and by beautiful flow cytometry, they found that there was decreased phosphorylation in all these pathways. So I wrote the clinical study to be able to do this in patients. I wanted to see if atorvastatin had activity in patients with low-grade or refractory non-Hodgkin's lymphoma. I set it up exactly like I described. I sampled the lymphomas by sticking a needle into them prior to therapy and day 8 of therapy, same time point as in the mice. And I wanted to determine if atorvastatin was really inhibiting the RAS pathway, which is upstream of all these molecules, and look at the downstream phosphorylation patterns to see if we're really impacting them, and then eventually correlate that to clinical response. And what we found and what we published in Nature Medicine, which was the first publication using nanoimmunoassay in clinical material, is we uncovered that there are decreases in oncogenic signaling indeed in patients that were treated with atorvastatin. So these you can see are peaks that are phosphostat5 and phosphostat3, and there's a dramatic decrease as early as day 8, which you can quantify. We can also do this by flow cytometry because in lymphoma, once we do a fine needle aspirate, you're essentially making a single cell suspension because they're very loose. And so we also did in parallel flow cytometry on the same specimens. And here I had to really crank up the differences because it's really hard to see a small difference. And so cranking it from -1 to 1, we can see that day 8, indeed, phosphostat3 and phosphostat5 did go down a little bit if we look at the CD20 positive cells, which are the tumor cells. And it was transient because day 43, when we resampled again, they were back to baseline. But the advantage of doing a single cell analysis is you can gate on both the tumor cells that are CD20 positive on the surface, but the CD20 negative cells in the sample were a lot of the immune cells, T cells, for example. And very interestingly, we could see that the signaling in these cells went up with treatment. So by using several different technologies, when you have the luxury of doing them at the same time, you can get high profile, a very high detail of phosphorylation for different phosphoisoforms, and then also start resolving the difference between tumor intrinsic differences and host, how the host is contributing to that. And just to really show you, though, how the nanoimmunoassay was really helpful for this particular study is when we did flow cytometry for phosphorylation of ERK, we really couldn't see much of a difference untreated to treated. But when we do the charge-based separation with nanoimmunoassay, we get very discrete differences that are highly quantifiable and measurable. So this is kind of showing you how complementary. One's really sensitive and specific, and the other one is really single-cell sensitive at that level. So this is another example of how we've used this in patients. Imatinib is kind of one of the first exciting targeted therapies, and it inhibits the ABL, BCR-ABL, and it inhibits the ABL enzyme and kinase, and it reduces phosphorylation of ERK. Now we know from literature, and many other people have shown that when you treat cells with leukemia with imatinib, that you see decreased ERK phosphorylation using just regular phosphospecific antibodies. So we did nanoimmunoassay on actual patient specimens of a patient with chronic myelogenous leukemia, CML, prior to treatment, and then again after initiating imatinib, and we found some surprising things. First, if you just look at the pre-treatment profile, you see a series of peaks, and by now, after Joanna's beautiful talk, you might recognize that most of it is unphosphorylated ERK2 and unphosphorylated ERK1, And this phosphorylated isoform, very interestingly, is a monophosphor ERK2. So the first surprise is that almost all of the phospho in this sample was in a monophosphorylated form, which is very different from what literature shows. Usually it's thought that you need both phosphorylations for the protein to be active. And if you had done this with just a Western blot, you see a band using a phosphospecific antibody, but with the phosphospecific antibody, you can't distinguish the difference between a mono and a dual phosphorylation. So you might assume it was all in the dual phospho. So first we found that it's all in the monophosphorylation. Second, because we can find out what percent of it was in the phosphorylated versus unphosphorylated form, we see that only 18% of it was actually phosphorylated. This is, when you've got all the oncogenic signaling pathways activated. After treatment, however, it basically went down to undetectable. It was less than 0.1% now in this phosphorylated form. And this was surprising because, it's 18% down to less than 1%. You might think you need a 90% inhibition or something. You have to start at 90% and go down to less than 50%. So this is actually showing us that it's useful to be able to profile things that are not necessarily highly phosphorylated, and even small changes, something that's low, not in an abundant isoform, but that disappears, that might be a very relevant thing to look at in patients. So we looked across a panel of patients with CML treated with the tyrosine kinase inhibitors, and it drives home a couple of more points. So first, if you look at all the untreated time points in blue from this series of patients, you can see that ERK is not highly phosphorylated. Again, that one patient was 18% here, but most of them were even less than that at baseline. So again, some of them were like 7% phosphorylation, and that's not something you would have predicted. The other point is, though, that there's a range, and it's not that they're all at 18% or they're all at 7%. They really do vary at baseline. However, what's very interesting is when you look at those that are treated, which are all, the same patient at its treated time point, you see that in the patients that respond, you're able to decrease the percentage of phosphorylation of that isoform. So in some cases, it's going from like 5% down to less than 1%. And that was a patient that responded, and it's something you really would have missed by almost any other technology because you just don't have that level of specificity and resolution. And then patients that were relapsed or refractory either went up or stayed the same. They didn't statistically decrease their levels. Now, this is also where being able to look at serial samples is really important, because if you had just looked at all the blue lines and you had your range, you'd be kind of say, okay, well, let's set a cutoff, and above this are the responders and below this are the responders. But if you set a cutoff like here, you would have missed these two guys that were responders. So being able to look soon after treatment is giving you much more discrimination to really see how patients respond based on individual response in the pathway. So I've showed you changes in phosphorylated ERK as well as phosphostat. And is it true in general that there are specific phosphoisoforms that seem to be preferred in other therapeutics? I showed you in CML that surprising result that it's a monophosphor ERK2. So we started looking at other targets in therapeutics, and with the goal of developing potential biomarkers kind of along these lines of the sequential change over time is really the biomarker, not just any initial level. So next I'll show you some data from that. So first, Picolog is a novel bryostatin analog, and this was in collaboration with the Wender laboratory, a chemistry laboratory here. So bryostatin, I don't know if you've ever heard of it, but it's a natural product that comes from a sea sponge. But unfortunately, it takes 4 tons of sea sponge to get 1 gram for use in patients. And there were clinical trials being run using bryostatin, which were quite promising in lymphoma. And the NCI had to shut them down because they just couldn't get enough to finish the study, let alone really think about developing a drug. So the Wonder Laboratory spent like 2 decades, because it's a really hard molecule to synthesize apparently, trying to make analogs that you could readily synthesize in a lab. And they came up with a lead compound, which they called Picolog, because picogram amounts in vitro seem to have some really good effects. So this was their most potent lead compound, and we did the first mouse study in the Felcher lab. So we gave this drug for the first time to mice with lymphoma, and lo and behold, the tumors actually had a response, and we were able to slow the growth of the tumors. The way the Picolog is supposed to work and the way the bryostatin is supposed to work is it activates the protein kinase C pathway. And downstream of that is phosphorylated MEK1. So in this case, we'd expect if the drug is effective and working through the pathway we expect, we see an induction of phospho-MEK1. So we should see the phosphorylations increase. So if you look at profiling the mouse tumor prior to treatment, you can see that phospho-MEK1, most of it's unphosphorylated. And there were 2 different phosphoisoforms, and they were 23% and 16% relatively phosphorylated. Well, after giving the drug, we saw that the unphosphorylated isoform went down, And it was preferentially this phosphoisoform that increased up to 42%. So again, we found something interesting, and that's that specific phosphoisoform seemed to be preferred even in the development of a novel therapeutic like this. So moving forward in the clinical trials, when this is ready to put in humans, we would want to be looking specifically at this isoform to see if that's what's being induced, to see if we can validate the target. Here's another example. This is a drug now called regurgitab, and it's a multi-kinase inhibitor, which basically inhibits polo-like 1 kinase as well as PI3 kinase. This was a clinical trial done here at Stanford by Peter Greenberg in hematology, and he enrolled 13 high-risk patients with myelodysplastic syndrome. These patients have a disorder of their bone marrow, and the bone marrow cells can't make adequate red blood cells, and the patients are anemic, and often they progress to full-out leukemia. So given these 13 patients that received this drug, 1 had a partial response, 8 had stable disease, which meant that they actually got rid of all the leukemic cells in their blood, and 5 progressed. And the median overall survival was 10 months, which in this population would have usually been 2 months. So there's definitely some activity of this drug in the patients. But we wanted to see what was happening at the molecular level. So what we did was we obtained CD34 bone marrow stem cells from serial bone marrow biopsies of these patients before treatment and early during the first cycle of treatment. And in the 5 patients, what we were able to get the cells from, we saw some very interesting things. If you look at the untreated in the blue, we looked at AKT2, which is a molecule downstream of PI3 kinase. So we'd expect that phosphorylation to go down. And what we do see is that if you compare the blue to the green, that the unphosphorylated form changed a little bit, but really there's a dramatic decrease in the phosphorylation of 1, 2, 3, and 4. And especially peak 3 really seemed to go down. So again, there's a hint that there's a preference for this phosphoisoform peak 3. And when we looked across, so when we look at this patient, we can see that the phosphorylation decreased and the unphosphorylated isoform increased. And if we look at all the different isoforms, we can see that it was peak 3 that really went down, peak 4, but some of them actually went up. So, so having this high level of resolution really allows you to figure out to what degree different isoforms are changing. When we looked across 5 different patients before and after, this is now graphed, minus 1.5 to 1.5, we can see that phospho-AKT2 actually decreased in patients that had the marrow response or stable disease, and those that have progressive disease, it actually increased. And most of that result was due to isoform 3, so that would be the key isoform to look at in ongoing clinical studies. So this was a single-arm phase 2 study. Now they've just completed the phase 3 study with multiple patients across the world, actually, and we're now starting to analyze the bone marrow specimens from these patients to see if we can validate this result in a larger cohort of patients, and if it really does predict response across the greater number of patients. So I think that's actually all I'm going to show you, so you'll end this session a little bit early. To summarize, the nanoscale immunodetection of proteins, I didn't talk about size-based, I focused on charge-based separation. These are enabling highly quantitative profiling of patient tumor cells before and during treatment. And profiles of individual protein isoforms and their relative abundance can be used to interrogate the degree of biologic response. And even a small degree of response could certainly still correlate to clinical response. So I showed you an example of atorvastatin in lymphoma. There's evidence of decreased STAT3 and 5 signaling. I showed you an example of imatinib, dasatinib in chronic myelogenous leukemia, where we saw decreased monophospho-ERK2, and that correlated with clinical response, as well as regurgitab in myelodysplastic syndrome, where we had decreased PI3 kinase signaling by AKT2 phosphorylation and stable disease. And in my preclinical work, these technologies like nanoimmunoassay are really making life a lot easier. I never run Western blots anymore. I just put stuff into the instrument and get my results the next morning. But for my clinical translational work, these technologies are really making the research possible. I mean, I'd been collecting fine needle aspirates on patients on clinical studies, but I didn't have a technology good enough to be able to really make measurements in them that I trusted. And now I can move forward with all this research. So in conclusion, nanoscale analyses, and Stanford is always developing all the time, as well as many other places, new nanoscale technologies beyond this even, to be able to be even more sensitive and look across a higher dynamic range with even higher resolution. So this is very much a work in progress. But nanoscale technologies are really helping with diagnostics and therapeutic monitoring for hematopoietic as well as solid tumors, and really enabling us to look here. But you can think with the ability to measure really small numbers of cells can help a lot of research, like looking at minimal residual disease. If we think, oh, are there a couple of cells left before we had no capacity to analyze them after treatment, and you can't see them on imaging, but if we can get these small populations of cells, now we can study them. Or even much earlier, I mentioned, something happens to initiate tumor, and you have to wait basically until the patient has a symptom or a mass that declares itself. Well, now if we know what to look for, we can analyze much smaller samples and really start pushing the envelope back to when we can detect tumors and do something about them sooner. So with that, I'll end with an acknowledgement slide that really this work takes a lot of people to do. Everybody from the surgeons that get us tissue to translational researchers, TRAM, as well as our lab members and collaborators and industry collaborators as well. So, and especially a big thank you to our patients, because they're the ones this is for. So if you have any questions, I'm happy to answer them. And I apologize that I didn't get you these ahead of time, but you will get the handouts.
Mm-hmm. It looks like there's a decrease in total ERK also.
Mm-hmm.
Is that where the decrease in phosphorylation is coming from, or is that an artifact? Or?
No, so that's a perfect question, because we can analyze both the total, like we do with the Western, and you normalize it to a loading control. And you can also normalize then the relative phosphorylation, which is a different measurement. And so both happened. Both happened. But it's not. But it doesn't entirely explain the relative percent change. Yeah.
So the other question I have then is if a lot of these readouts, I guess I would say, are not the direct effect of the drug, it's like more
Downstream. Mm-hmm.
Do you have any intention of like trying to combine them with
Absolutely.
Like a mechanism
Oh yeah.
or an APC?
Absolutely. This is exactly where it's most useful. So, and ideally, not only we don't want to just look at the direct target and downstream, but also compensatory pathways to really get a sense of what's happening. So yeah, the more that we can profile, the better. Go ahead.
Yeah, I found it was a very nice example how you can apply this technology to patients. I was just wondering how you determine what time point to choose to look for your target inhibition.
Yeah.
Because I assume that the inhibition of phosphorylation or upregulation precedes the clinical response.
Mm-hmm. And that's kind of the entire point of why we want to look much sooner, but you're absolutely right, because it depends on the half-life of and steady state of when the drug gets there and when you really think. So for the statin study, I was lucky to be able to know in the mice we did all the different time points, and we saw day 8 was a good one. But luckily in patients, because we can now, if we can get away with this whole minimally invasive sampling, either by fine needle aspirates or ideally blood draws, you can do multiple time points to see when you see the maximal effect. And really the time to do this is when you're doing the phase 1 studies, when you're actually doing these multiple samplings of the patients and really getting a close kinetic analysis. So what I'd like to really build into studies is a kinetic analysis of the target, not just of the blood levels of the drug. So, yeah.
Are you looking for targets in peripheral blood as a substitute for tumor biopsies? Because that would be much easier.
We're looking at everything. Absolutely. So right now, I haven't published yet, so I didn't present it, but we're looking at circulating tumor cells before and at multiple treatments, time points during treatment. We're also just looking at serum proteins, because you could certainly use this to look at that, although that's been a little bit harder because there's so much other signal that kind of gets in the way in serum, as well as looking at different immune cell populations from the blood and or tumor. So lots of things to look at, absolutely. Okay, thanks.
So let's take a 10-minute break, and then we'll start back with